Hello and welcome to episode 11 of the series. And today's fragrance is Aqua Sextius by a small French company called Jules et Mad. And it was composed by a very talented young perfumer called Cécile Zaroquian. And I find it absolutely wonderful because it's a deceptively fresh and simple and transparent fragrance, but actually it has tremendous staying power and radiance. And it's a very hard thing to do both. Usually when you have a fresh fragrance, it goes away quickly, and when you have a big heavy one, it's not fresh, but this seems to combine both. So first of all, let me apologize if today you can hear a background sound which is not normally there, and that's because I have to run a fan because it's so hot today in Athens that I simply find it impossible to speak into the camera without sweating profusely. So the purpose of the lecture today will be to try and understand ultimately what the most important difference between a shape theory and a vibration theory is. And if I can sum it up as simply as possible, it is the following. Imagine that you have a set of receptors. You have, say, 10 receptors. And the smell of a molecule is determined by its shape. So each receptor is wired to a particular part of the brain. And when a molecule comes along, it binds to receptors, let's say 1, 3, and 9. And that 1, 3, 9 is the actual pattern which determines what the molecule smells of. Suppose now that somehow I can change the shape, not of the molecule, but of the receptor, so that I change all the shapes of the 10 different receptors by changing one particular amino acid in the protein, and now they will bind to a different set of molecules. So I come back with the same molecule, which had 1, 3, and 9, and now it binds to 2, 4, and 7. That means the smell of that molecule will have changed. It will be different. So how is vibrational theory different? Well, vibrational theory says that the smell is actually written in the molecule itself, such that the vibrations of the molecules, which are quite independent of whatever it binds to, are what determines the smell. So if you have a sufficient number of receptors that will bind it and that will analyze its vibration in a particular frequency range, then you can change all the shapes of the receptors if you want. What happens is that it'll bind to receptor here, one receptor here, and one receptor there, and then you change them, and it'll bind to a different set of receptors, but always the vibrations will be probed in the same way. So in other words, the smell of a molecule is written into the receptors in a shape theory and into the molecule itself in the vibration theory. This is a very fundamental difference. So let me illustrate this with some drawings to make it clearer. On the left here I have 3 shape-based receptors, and the binding pocket is drawn to have 3 different shapes to which the odorants should bind. And on the right-hand side I have the idealized vibrational receptor, which is a completely featureless binding pocket, here represented by a square with round corners. Now, this is of course an idealization because the binding pocket of any protein will always have some degree of shape. It cannot be like a cuvette that you put in a spectrometer, which is much, much larger than the molecule, and therefore its shape doesn't matter. In the case of a receptor, the binding pocket is always, by definition, the same size as the molecule it'll bind to, and therefore it must have some sort of shape. But here I've drawn the ideal situation in which the receptors, the vibration receptors, have no shape preference. They're basically completely unselective, whereas the shape receptors are selective for a particular shape. So now I put in an odorant with a particular shape, and what you see is that on the left, 2 out of the 3 receptors do not bind that odorant. The third one binds it, whereas on the right, all 3 receptors bind the odorant. So what happens next? Well, in the case of a shape theory, on the left, the receptor that binds the odorant is turned on, so that you get a one, so to speak, a bit of its response is turned to 1. And on the right-hand side, for the vibrational theory, only one of the receptors is tuned to the correct vibrational band, and so that particular receptor here in red responds to the vibrations of the red odorant molecule and is turned on, and the others, although they bind it just as well, are not turned on. And that is one of the basic mechanism by which each theory actually works. On the left, binding is the event that turns on the receptor, and on the right, binding is only one of two events that turns on the receptor. The molecule also has to have the correct vibrations. Now, what happens if I change the shape of the receptors? So that, for example, in the wiring, all of a sudden, the receptor that binds the odorant is no longer wired to the brain in the same way, but it's been moved. That shape is now corresponds to a different wire. Well, in the shape theory, that means that the smell will be different. We've gone from a binary word 0, 0, 1 to a binary word 1, 0, 0, so that the smell, perceived smell of that particular odorant will be completely different. In the vibration theory, of course, in the rather idealized limit of completely non-specific receptors, it'll make no difference because the wiring is by frequency band, and as long as there's a receptor that binds to that particular odorant that has the correct frequency band, then it'll give the same smell. So I've just explained that in the case of the idealized 3 vibrational receptors, I had to assume that the binding pocket was completely non-specific, which is completely unrealistic. It always has some degree of specificity. So in the case where we only have 3, let's say, vibrational receptors, one for each frequency band, let's say, of course this thing wouldn't really work, because if I changed the shape, it would actually affect the binding. But the situation that we have in reality is quite different. We have lots of receptors, 400 in man, 1,000 in mouse, etc. So you can have a situation in which you have enough receptors in each vibrational band to make sure that if the shapes are different, nevertheless, the same, on average, the same amount of activation of a particular vibrational band will occur. And this is depicted in this slide, where you have the 3, let's say, we don't know how many vibrational bands there are, but let's just assume that there are 3. The red, the green, the blue, and each of these categories of vibrational frequency bands has several receptors, here depicted as 3, and each band therefore has different shapes of the receptor. Now if I put odorants, what will happen is it'll bind more or less, but it'll bind pretty much to all of them, and it will turn on only those receptors that have a sensitivity to a vibration of the odorant, and that looks maybe something like this. In the red vibrational band, one receptor will be turned on 2.7, one to 0.6, and one fully to 1. And the idea is that ultimately the exact strength of binding of the odorant to that particular receptor will determine how efficiently it's turned on, but that overall you will get a pattern that says only the red receptors are turned on. The odorant is bound to the others as well, but because they have the wrong vibrations, they're not turned on. So in this particular case, and if you expand the number of receptors from 3 per band, depicted here, to, let us say, I don't know, 50 per band, you can arrive at a situation where no matter what odorant comes along, it will on average result in the same activation pattern per band, so that it doesn't matter whether it binds much more strongly to this or that receptor. It will always give you the same rough pattern of activation of receptors per band. So in other words, an odorant which has some red vibration but no green and no blue will always give you mostly activation of the red band and none in the green and blue, and therefore will give you the same odor character. So you might say, well, since changing the shape of the receptors will answer the question as to whether we're dealing with a vibration or a shape sensing system, why don't we change the shape of the receptors? And the answer is, well, it's not that easy, but it's not impossible. What you could in principle do, but not in practice, is genetically engineer each receptor such that the binding pocket is changed in shape. The reason you can't really do that, apart from the very large number of receptors, is that we don't even know which amino acids of the protein, receptor protein, are actually involved in the binding of the odorant. So there's a double difficulty there, which basically prevents that kind of experiment from being done right now. However, there is a way of doing this which is kind of clever and can answer the question somewhat indirectly, but is doable. And that is the following. I've told you before that humans have 400 receptors and flies have 63. I've also told you that the fly receptors are completely different in genetic sequence and in fact even in the orientation in the membrane to the human ones. So in essence, the fly receptors are, if you want, a set of smell receptors that are completely different in shape from ours. Now, how do we find out if the flies smell the same things we do? So from everything that I've told you so far, you can guess that some of my favorite reading would have to do with molecular vibrations, and you'd be right. And so I'd been looking for a long time at a book that I wished that I had early in the first year when I figured the basics of the vibrational theory out, because it would have saved me an enormous amount of time and trouble. But even having bought this book, which is absolutely a wonderful reference book in the field, it took me a long time to notice something really remarkable. You know, and I've told you before, that nitriles have both a peculiar vibrational frequency at 2150, 2200, and a peculiar smell. But even more interesting is the fact described in George Socrates' book that there's a type of nitrile which has not only a CN group, but right next to it OH group. And the presence of the OH group makes the nitrile invisible to a spectrometer. So here you see the CN stretch of the 2-hydroxybutyrylnitrile, and you can see that it's just under 2 divisions of intensity. And you also notice the surface around the molecule is the electrostatic potential, meaning the charge on the different parts of the molecule. And you can see that there's a small red cap which indicates negative on the nitrogen of the nitrile. And this part of the video is the 3-hydroxybutyrylnitrile, and you can see that the CN stretch peak is now more than 3 divisions instead of 2, and that the red cap is much larger, indicating that the charge on the nitrogen is much bigger. And the conclusion one draws from that is that what happens is that the position of the hydroxyl group changes the charge distribution in the molecule in such a way that when it's on the carbon next to the nitrile in the 2 position, it redistributes the charge so that you can no longer see the CN stretch. You need charges on it to be able to see it. Whereas when it's on the 3 position, it's simply too far to do that. And therefore, you smell the nitrile again because the charge hasn't been perturbed. So basically, the 2-hydroxy compounds have no nitrile vibration detectable in the spectrometer. And the 3-hydroxy have one, and both of them can be ordered from chemical suppliers. So we ordered both, and to our delight and surprise, the 2-hydroxy had no nitrile smell. You absolutely couldn't tell that a nitrile was present in the molecule, whereas the 3-hydroxy did have a nitrile smell. So, first of all, that's very good news for a vibrational theory, since, after all, both of these molecules have the same functional groups. They have 4 carbons, 1 hydroxyl, 1 nitrile. The main difference is that in one case, the vibration is not perceived by either a spectrometer or our nose, which suggests that our nose may be a spectrometer. But this also allows you to do a really remarkable experiment, which is to say, do flies feel the nitrile the same way we do? Suppose that flies do not smell the 2-hydroxy compound, but smell the nitrile in the 3-hydroxy compound. This can only be explained by vibrations because the receptors in flies are completely different from ours. So even if, by some extraordinary coincidence, in the case of our receptors, this could be explained by some weird thing to do with shape, it would be practically impossible to imagine that the same coincidence happens also in flies. So how do you test this? Well, it's quite simple, actually. You train them against a nitrile, as we did before with these citronellyl nitrile and the aldehyde. And then you ask, when trained against the nitrile, do they avoid the 2-hydroxy? Can they smell the nitrile in there? And do they avoid the 3-hydroxy? And the answer is, as you can see from the illustration of one of the figures in this paper, that Cleo Maniati and collaborators did and published in the eNeuro. The flies, when trained against an aldehyde, pretty much don't understand the question. When trained against the nitrile, they avoid the 3-hydroxy, which is the compound that we can smell a nitrile in as well. So this is really quite strong evidence for the fact that flies smell things the same way we do, and taken together with the fact that we seem to smell molecular vibrations, they seem to smell molecular vibrations, they have very different receptors from ours, that the same mechanism is at work in both cases, even though the receptors are evolutionarily rather different. So this is very encouraging. Another experiment which is described in that paper, which is also really interesting, is the question, do flies smell boranes the way we do? In other words, do they smell them like sulfur? Now how you test that is you expose them to boranes and to some completely unrelated compound and to a sulfur compound. And you ask, if I train against the unrelated compound, do they avoid the borane? The answer is no. But if I train them against a sulfur compound, do they avoid the borane? The answer is yes. And vice versa, when you train against boranes, they avoid the sulfur, but not some other thing. So it's not just that they dislike a smell, it's the specific odor of the boranes. Interestingly, in flies, you can completely shut down their sense of smell by a manipulation of one particular protein, which is essential to the sense of smell. So you can check, in all these cases, that they're not simply reacting to some sort of unpleasantness in the stimulus. After all, boranes are quite toxic, so you could imagine that they would dislike boranes or avoid them for very good reasons. This has nothing to do with it. In fact, when flies are deprived of their sense of smell by removing this particular protein, then they no longer respond in this fashion. So we know it's a sense of smell. We also know it's not taste, because the taste is not affected by this protein. So in conclusion, we can pretty much say at this point that the evidence from flies taken together, the fact that they can smell isotopes, the fact that they confuse nitriles with deuterium hydrogen because of the similarity in vibration, the fact that they smell nitriles the same way we do when the nitriles are cancelled out by hydroxyl groups, the fact that they smell boranes the way we do, namely sulfuraceous. All of this really pretty much suggests that not only is the nose a spectrometer, but it's also a spectrometer that has been used several times by evolution, since the fly receptors are different, completely different from ours, but yet seem to respond in a similar fashion.